Now, as we saw in a previous chapter, you want to study bacteria? No problem. You pull out an agar plate. You streak across it with an inoculum from some swab you've taken somewhere. Or you dip the loop that you're transferring the inoculum into a broth. Wait 24 hours, got all the bacteria you could ever want. Well, viruses are a bit different. I could take a vial that has 1,000 viruses floating in a solution. I come back a couple of days from now, come back a couple of weeks from now, months from now. That vial will still only have 100 viruses. Viruses cannot replicate on their own. Viruses cannot increase their population number on their own. So, once we've discovered, hey, there's these things called viruses, we have spent the past century trying to come up with ways, mechanisms in the laboratory to study them. To study them, you have to first be able to propagate them. You have to be able to go from, look, I've got a few that I've gotten from a patient sample. And then you have to be able to go from just a few to thousands or millions of them, so you have enough to start running tests with. So, purposes of viral cultivation, as you see in your textbook. Isolate and identify. Come on. Koch's postulates, or Koch's postulates. Koch's postulates apply even to viruses. It's just a little harder with viruses, because first, not only do you have to be able to take it from a person, but you have to be able to find a way to grow it in the lab. Postulate number 2. An appropriate means of growing it in the lab. Once you've done that, then you can go on to postulate 3 and 4. As they're finding right now with COVID, trying to come up with vaccines, well, you have to have a way to take a small sample and exponentially increase the numbers. Because a vaccine's not going to work if I come around and I just inject one virus into everybody in the city of St. Charles. That's not enough. You need more. And to be able to have more, you have to have a way to propagate, increase the numbers. And then we have to know exactly how to mutate it so that it's not the killer. Do we give you the entire virus just mutated so it's not a killer? Or do we break it down into its pieces and then inject you with just the pieces of the nucleocapsid? So how do we propagate? Well, live animals. If we understand what the host range is, I'm not going to do this to humans because we're not Nazis. That was done during World War II by the Japanese and the Germans. Never again. So we go with animals. Animals that are close to us, genetically speaking, metabolically speaking. So that way their receptors are quite similar to ours. Unfortunately, you're going to have to go with the great apes, the primates, the monkeys. They're very close. We're begin looking into genetically modifying pigs. Pigs are close, to us anatomically. It's just at the cellular level to where the problems come about. So if we can alter their genetics so at the cellular level they're closer to us, well, maybe we're onto something. If all we want to do is just propagate enough viruses that have already been studied, already been mutated, so it's no longer deadly, well, turns out bird embryos, chicken eggs, are the best way to do it. You take the viral sample, the viruses that's in a solution, and you punch a hole, a small little pinhole through the shell of the egg of a chicken egg that's only a few days old. Embryo still there, hasn't started growing too much, it's just a fertilized egg. You inject it into the yolk sac. As the chicken develops, starts to grow, it'll become infected. One virus goes into a chicken cell, hundreds, thousands come out, spread to the neighbors, and it just keeps going and going and going. That's why if you ever go and get a vaccination most of the times, they should ask you, are you allergic to eggs? If they ask you if you're allergic to eggs before they give you an injection, that means that virus, that vaccine strain of the virus, was propagated in a chicken egg. Unfortunately, when they go to isolate the viruses, sometimes they bring along egg proteins, which is why some people later in life, after getting vaccinations, develop an allergy to eggs. They got too much of the chicken proteins injected into them, and now their immune system recognizes it and mounts an attack whenever they try to eat something with eggs. Not all egg allergies are from that, but quite a few are. And then there's tissue culturing. We take cells, human cells that are cancerous, they're constantly growing, and we put them in tissue culture flasks. As you look at the pictures in the textbook, you'll see that we can easily do this. Problem is, it's very labor intensive. This is great for early research, but once you've gone through the early research, we know what the DNA is like, we know what the proteins are like, and now we want to study the cytopathic effects. Well, then you have to go to live animal. Once we've studied, here's the disease course, now we need to come up with a vaccine. That's when you go to the chicken embryos.